Great. So our next speaker is Andreas Hanke, who's going to talk to us about denaturation. I can't say it. Denaturation transition of stretched DNA in the presence of DNA binding ligands. Andreas.
Thank you. Okay, this is her work. I do in collaboration with a group in Northeastern University, Laila Chokri, Yuna Rosina, and Mark Williams. They are doing this stretching experiments in the presence of DNA binding ligands, and I do some of the theory. Okay, first DNA. Of course, we know DNA is a double helix held together by base pairs. So if we look at it more closely, is there a pointer somewhere? Okay. If you look at the inside of the DNA, the two strands are held together by base pairs, AT and GZ base pairs, and the energetics is also determined by base stacking. So this was a discovery by Watson Crick, and it was actually made very close to here at the Eagle in Cambridge, 1953. So we can really go to the footsteps, so to say, and follow the discovery to the same place where the discovery was announced. In the last 15, 20 years or so, has been big progress in force probes of DNA. So it is possible to probe the mechanical response of single molecules. And one possibility is an optical tweezers instrument. And this is the type of instrument that the group around Mark Williams is using. So there is a long DNA molecule, like lambda DNA, is attached between two spheres. One of the beads is trapped in a laser beam. And it is possible to detect the force as a function of extension by measuring the deflection of the bead that is trapped by the laser beam. So in this way it was discovered by various groups, Gauck and Bustamante and others, that DNA exhibits a very characteristic behavior when you stretch it. There is so here's the extension and here's the force. And so when you start extend DNA, then first what happens is kind of expected. It follows the theoretical curve for worm-like chain model corresponding to double-stranded DNA. But then at about 65 piconewtons, something unexpected happens. There's a force plateau. That means you can extend the DNA about quite a bit relative distance at about 65 piconewton. So the force doesn't change, and only the extension changes. And only at a much larger extension, there is again an increase in the force. And this time, the force extension relation is described by a worm-like chain model, this time corresponding to single-stranded DNA. So there are various explanations or attempts of explanations of this phenomenon. One explanation is that this overstretching transition corresponds to force-induced melting. That means as we go along the overstretching plateau, more and more denatured regions occur, or denaturing bubbles. And so in this way, you have a transition from double-stranded DNA to single-stranded DNA. At the end of the overstretching transition, you have basically two separated strands that are only held together by GC-rich regions. There's some debate about this models, so there are competing models, for example, sDNA or nicked DNA and so on. And for that, I refer to the poster by G.A. King. It's outside to see some of this debate, but we will follow here this model that's also the group by Mark Williams is following. So we interpret the overstretching transition as force-induced melting. So the traditional approach to DNA melting is done by heating. And so here the melting transition occurs at a temperature at about 80 or 85 Celsius. And there we observe a transition from double-stranded to single-stranded DNA. And quite analogous, now force-induced melting instead of temperature, we have force. And the good thing is we can do this at physiological temperatures and not at elevated temperatures where proteins, for example, would decay. And that is important for experiments where we probe interactions of DNA with proteins. So we can, in a controlled way, induce a partial or complete denaturation transition, and in this way probe the interactions of ligands with single-stranded DNA and double-stranded DNA under a wide range of conditions. And this is what Mark Williams' group is doing. So in this talk, we focus on a particular kind of ligands, namely intercalators. And in particular, I look at an experiment done on the intercalator ethidium bromide. Ethidium bromide is a small molecule that binds to double-stranded DNA by intercalating between adjacent base pairs. It is a common fluorescent tag to detect nucleic acids in the lab, and it affects, obviously, DNA replication and transcription because it intercalates between double-stranded bases or double-stranded DNA. It is also a possible model for anti-cancer drugs because of its activity to inhibit DNA replication. So in this way, prohibit or inhibit replication of cancer cells. So again, an intercalator binds between adjacent base pairs, and in this way also elongates the DNA. So to say, the intercalator goes in between, and in this way the DNA gets longer. This, of course, is important for the stretching experiments because we probe force extension relations. These are experimental data made on ethidium bromide under various concentrations, different concentrations of the intercalator. So for example, the black curve is the force extension relation in absence of the ligand, corresponding to the curve that I have shown in the beginning. The usual overstretching plateau at about 65 piconewton. Then as we increase the concentration, the plateau occurs at higher force and starts later, but finishes more or less at the same force. So that's the characteristic features when we increase the concentration of the intercalator. It's a higher stretching force and then starts later, the overstretching plateau. It makes sense because, like I said, the intercalator elongates the DNA, so to say it makes it less favorable to melt because it's already, double-stranded DNA is already longer. So in this talk we try to explain or to say to derive these stretching curves from a theoretical model and to reproduce these curves with our theoretical model. So one aspect of the model that I present is it has a multi-scale nature to it. What does that mean? We try to explain stretching curves on a DNA molecule that is about 50,000 base pairs long, so it's very long. On the other hand, we have very nice and accurate data for short DNA pieces by various methods. For example, molecular dynamic simulations we have seen in various talks. So we have already quite precise information for short scales, but how do we translate this or elevate this to the long length scales relevant for these stretching experiments? For this, we propose a model where we can incorporate information from the short scale into a model that then works on long scales. And this way it corresponds to a multi-scale modeling. So the way this works, it's the simplest version, is a polar-Charaga type approach. So here is the DNA molecule, here modeled as a sequence of denatured bubbles and bound intact DNA. And the pink dots correspond to the intercalator that can bind to the double-stranded DNA. This intercalator just binds to the double-stranded DNA, so to the green parts. Green corresponds always to bound DNA, and red indicate then denatured bubbles. So what is given here is the concentration of the intercalator, and then the model also is described by various statistical weights corresponding to a denatured loop, internal loop, to a bound segment with intercalating molecules, and also to an end region. And then also another control parameter is, of course, the stretching force F, with which we pull the sphere to the right. The basic idea of the model is actually quite so mathematically quite nice. It also incorporates the, so to say, the topology of the different sequences that you can form here. So in this case, what we have, we build the grand canonical partition function in an ensemble where the force is fixed, where the fugacity is fixed, the fugacity that's conjugate to the number of base pairs, to the total number of base pairs. And then this partition function obtains as from the corresponding canonical partition function by integrating over all extensions conjugate to the force and summing over all number of possible base pairs conjugate to the fugacity. And we can express this partition function as a series in various statistical weights that I indicated here. So this series indicates that we always start on the left with an end region, and then we have various powers of insertions of a bound region and an open loop region. So for example, you can start with 0, then we just have open end, bound, open end. The next term would be open end, bound, internal bubble, bound, open end, and so forth. And the B and omega terms include the complete statistical weight for all possible combinations of a bound piece and a denatured loop. Then you can sum the series and you get a compact expression for the total partition function in the grand canonical ensemble. So that's an advantage that we go to the grand canonical ensemble. It's just a geometric series. So the statistical weight for a bound segment obtains in the following way. So let's assume that we have available the free energy of a bound region, for example, from molecular dynamic simulations. We assume that it's extensive. That means this free energy is proportional to the number of base pairs, and AdS is then the free energy per base pair. We can go over to an ensemble with fixed force via Legendre transformation, and in this case, the ingredient would be, or the outcome would be the Gibbs free energy per base pair for fixed force. Then the extension obtains as a derivative of this free energy for fixed force as a derivative over the force. So once we have the free energy per base pair, we go over to the grand canonical ensemble by introducing a fugacity and chemical potential of base pairs. And for technical reasons, we also introduce fugacity and chemical potential for bound base pairs, denoted with mu B and B. So then we build the grand canonical partition function. We can sum it like before. We get a compact expression for the statistical weight of a bound segment. And the ingredient here is the Gibbs free energy of a double-stranded piece of DNA, which could be obtained by a molecular resolving model. Now, if there's also an intercalator, then what happens now? So let's repeat a bound segment is formed by 1 base pair, 2 base pairs, 3 base pairs, and so on, and summing over those. Then we get the series. Now, if there's an intercalating molecule present, then we have combinations like those. So B is now the complete statistical weight for all bound states without intercalator. Then there can be 1 bound, there can be intercalator separated by a bound segment, intercalator, and so on. Then we can sum this as well in the same method, and we get statistical weight now for a bound segment including intercalator, and the control parameter I includes the Gibbs free energy of the intercalator and the fugacity of the intercalator, which is related to the concentration in solution. Okay, for the internal loop, we do quite the same thing. So everything is replaced now. Instead of double-strand, we have single-stranded quantities. For example, here, the Gibbs free energy per base pair for single-stranded DNA, but the rest is analogous. And once we have these contributions, we can incorporate this in the partition function. This was the expression for the partition function. Here we incorporate now the statistical weights that we just have calculated. And like I said, the bottom ingredients are the Gibbs free energies for double-strand, single-stranded, and with intercalator. But these quantities are in principle could be simulated in simulations, molecular dynamic simulations. Now, once we have the partition function, you believe me that we can do thermodynamics. We go over to the thermodynamic limit by controlling this fugacity. We have to choose the fugacity that we have that reach the thermodynamic limit. And now there's a nice thermodynamic trick. So what we look at is the mean fraction of bound base pairs. So that's the ratio of the mean number of bound base pairs over the total number of base pairs. And we can also look at the mean DNA extension per base pair. That's the mean extension over the mean number of base pairs. And now we used the Gibbs to Hem relation for the partition function that acts like a potential of the force, fugacity, and fugacity of bound base pairs. So if we do that, then we can deduce these fractions, mean fraction of bound base pairs, directly as a function of this critical value of the fugacity that corresponds to the thermodynamic limit. So we need to have a derivative with respect to b at b equal 1. And for the extension, we follow the same trick. Okay, now if we do that, we come to the results. The first result is the mean fraction of bound base pairs here without the DNA binding ligand. For different values of a parameter that is called the cooperativity parameter or loop initiation factor, that's the statistical weight that punishes the formation of loops. So in experiments, typically values are used that are about 10 to minus 3 of this parameter. And we see that we obtain a more or less sharp transition. So what is varied here is a parameter omega, and omega gets larger when the force gets smaller. So varying the force varies omega via this expression, and then we get a sharp jump of this fraction of base pairs from 0 to 1. The ingredients of this omega parameter are the melting free energy per base pair and the increment in DNA extension per base pair. Everything can be derived very cleanly by this model. So there is no hand-waving arguments. That's really cleanly derived by the Polan-Charaga model. It comes out like that. And now finally to the comparison that I promised. Remember, I showed you the plots with experimental data, force extension relations in the presence of the intercalator for different concentrations. And now using our model, that's the corresponding curve. I have used the same colors, and for different values of the statistical weight of the intercalator. Remember, it depended on the concentration via the fugacity and also on the free energy of the bound intercalator. So I have what I adjust here is not much, actually. I adjust the values of this parameter. I cannot calculate it, but I can adjust it so that I get corresponding curves. And I also choose the experimental result for the extension of a bound base pair and the extension of a unbound base pair directly from the stretching curve. But I ignore the dependence of these two quantities from the force. So these are the only three parameters that enter. And I choose for the cooperativity parameter the value 10 to minus 3. So that is this value here. And so you see that the basic features are actually reproduced quite well. I get the overstretching plateau for the case without intercalator, and I get the same qualitative behavior. So for increasing concentration, increasing I, the force increases, and also the plateau starts later at higher extensions. So outlook of this model, the simplest version, but it's a possibility to incorporate information from atomistic simulations, for example, microscopic information, into a model that then elevates to high length scales relevant to experiments. And I think it's promising. It works quite well for this very first preliminary version. We're working on it and also doing experiments with other molecules. So it is still ongoing work, but it looks promising. Thank you very much.
Just a very quick question while Jakob sets up.
But it couldn't be physically vertical, right? I mean, when the force goes up, then you can keep extending it.
Yes, so that's right. So I should say here, this curve is only valid really for the overstretching plateau itself. I have not included the parts where we have completely single-stranded DNA. You would then describe it again by a worm-like chain model, for example, for single-stranded DNA. So this vertical rise is not right, so it is, so to say, an artifact that I just consider as overstretching plateau. For example, these dotted lines or dashed lines are actually correspond to a worm-like chain for double-stranded DNA, but I didn't do the same thing for single-stranded DNA.
Okay. Thank you very much. Thank you, Andreas.